Well, it's a pleasure to be able to present today our work in the use of mRNA for treatment of cardiac fibrosis. And before I begin, I just want to declare my conflicts as having equity in a new biotech company called Capstan that's focused on bringing these therapies forward to the clinic. So our interest in using engineered T cells to try to treat cardiac fibrosis really was spurred from some publications such as this one, which showed that you could ablate activated cardiac fibrosis in various mouse models of heart failure using genetic tools like expressing diphtheria toxin using a Cree driver mouse. And the result was not only elimination of those activated fibroblasts, but reduction in overall fibrotic area, meaning the extracellular matrix was getting resorbed, and also an improvement in function, as measured here with the ejection fraction. So we began to think about whether we could come up with a more translational approach that we could use in people to ablate activated cardiac fibroblasts in the heart. And we did that by looking for cell surface markers that we could target with engineered T cells using chimeric antigen receptors. And you may be familiar with this approach because it's been so successful in cancer, especially in leukemia and lymphoma, where a modified T cell receptor type structure known as a chimeric antigen receptor specifically targets an antigen on the surface of a cancer cell. And we asked if we could identify such a unique antigen on the surface of activated fibroblasts in models of heart failure. We did that by starting with human heart specimens, which our colleague Ken Margulies has been accumulating from patients undergoing cardiac transplant or having biopsies of their heart. And we analyzed RNA-seq data sets from patients with either hypertrophic cardiomyopathy, HCM, or dilated cardiomyopathy, DCM, for genes likely to come from fibroblasts that were expressed only in the diseased hearts, but not in normal hearts. And the very top of that list was a gene encoding a protein called FAP, fibroblast activation protein, which you can see listed here. And interestingly, some of the other genes that came up on this list are also known to be expressed in activated fibroblasts, including, for instance, periostin. And this gene is what had been used as the Cree driver in that experiments that I began with using diphtheria toxin to kill activated fibroblasts in the heart. So we had some confidence in this gene list. And we looked to see whether FAP, the protein, was expressed in human hearts. And sure enough, we see it in the interstitium here and between the myocytes by the activated fibroblasts in hypertrophic cardiomyopathy or dilated cardiomyopathy, but not at all in normal hearts. It's not expressed on quiescent, non-dividing fibroblasts that are present in the normal heart, only in those that get activated in disease. And we've gone on to look at a number of different examples of human heart failure, sarcoidosis, scleroderma, giant cell myocarditis, et cetera, including COVID-19 myocarditis. And we see FAP expressed on these activated fibroblasts in all of these conditions. So we think it's a pretty interesting target to go after. Now, what is FAP? It's a cell surface glycoprotein. It's a gelatinase in the serum protease family. It's expressed during embryogenesis in normal embryos, but not in adult tissues unless they're injured or in cancer. And it's only on these activated, proliferating fibroblasts. It is on tumor stroma, and in fact, it's been targeted in some cancer therapeutic approaches. And it's been seen in the human heart, especially in scar after myocardial infarction. So we turn to a mouse model of heart failure that's commonly used where a implantable pump delivers angiotensin II and phenylephrine to induce essentially hypertensive heart failure. And you can see that just after a week of this pump in place, there's extensive fibrosis in the myocardium of these hearts. And to test the idea, we delivered FAP CAR T cells engineered with retrovirus outside the body and injected intravenously back into these animals, first at 7 days and then at 14 days. But recall that at 7 days, this fibrosis already exists. So we're trying to treat existing fibrosis, not to prevent the onset of fibrosis. And the results in this experiment were really striking. First, we could see that a GFP-labeled CAR T cell found its way into the fibrotic regions of the damaged hearts, but not in the interstitium of the normal hearts. And after 4 weeks, the extensive fibrosis seen in these treated hearts was almost entirely resolved. The type of experiment you only hope to get once in a while, the type of result, sitting at the microscope, I could pick out the treated slides, right after the first experiment. You see this extensive reduction in fibrosis, and along with that, both systolic and diastolic function was improved in these animals. So this work was published a little while ago, and it received a lot of attention. There was commentary in the New England Journal and Scientific American, et cetera. But we still weren't convinced that this was going to be the best therapeutic translational approach, because normal CAR T cells made as we made them in this experiment last forever in the body in humans. And some scarring is good. If you have a heart attack, you want to be able to scar that region of the heart. Or if you cut your skin, you want to be able to scar the skin. So we thought it would be more appropriate if we could develop a transient CAR T cell that would only have an effect for, let's say, a couple of days. And for that, we turned to the idea of making our CAR T cells with mRNA instead of a retrovirus, perhaps mRNA modified to be stable and encapsulated in lipid nanoparticles, exactly as is done with the COVID-19 vaccines. We began this work collaborating with Drew Weissman, who did the foundational work for the COVID vaccines. We began it before COVID existed, but it turned out to be a great partnership. So mRNA can produce transient CAR T cells. The CAR is only expressed on the surface of the T cell for as long as the mRNA lasts in the cell. You could titrate the therapy, you could dose it in patients, and we think would have reduced risk compared to a long-term T cell for this purpose. Turns out lipid nanoparticles can be targeted specifically to T cells by decorating them with an antibody that sticks to the T cell. And this provides a potential for in vivo CAR T engineering. Maybe you don't have to make the CAR T cells ex vivo and inject them back in. Maybe you could just inject these mRNA lipid nanoparticles into the body and have them engineer the T cell in vivo. And that would avoid the expensive ex vivo manufacturing step and allow it to be a scalable therapeutic. So here's just a diagram that shows our decorated lipid nanoparticle being endocytosed by a T cell, where the RNA is expressed and the chimeric antigen receptor expressed on the surface of that T cell. And this would obviate the long, complicated manufacturing process now in place for making T cells for patients with cancer. So we first packaged the LNP with an RNA expressing luciferase and showed that it could be targeted specifically to the spleen, where T cells are found in the body. LNPs are also taken up normally by the liver, as they are with the vaccine, but you can see how specific the uptake in the spleen is only when we target those LNPs with an antibody on the surface. And if we mix those antibody-coated LNPs expressing a FAP CAR with T cells in a dish, we see about 80% of the T cells take up the RNA and express FAP on the surface. And when we inject those lipid nanoparticles directly into the bloodstream of a mouse, we see that about 20% of the T cells in the spleen will express FAP on their surface. So we can effectively deliver this RNA directly to T cells. Now, the amount of FAP CAR expressed on the surface is not quite as much as we see on the surface when we make CAR T cells with retrovirus. So it's not quite as dense, and we might be able to optimize it further. And that FAP expression lasts only a couple of days. You see it quickly decay here over a week. So we are indeed making a transient FAP CAR T cell. But when we deliver these to injured hearts, we can find them in the myocardium where fibrosis exists. Here we're staining for the FAP CAR on the surface of T cells and CD3 positive T cells, and you see them here found in the fibrotic regions of these hearts. And in the same heart failure model I described previously, using angiotensin and phenylephrine, we were able to show that we could both reduce fibrosis and, once again, improve both systolic and diastolic function in this animal model. So we could make an effective CAR T cell in vivo using modified RNA and lipid nanoparticles. So finally, I'll close by saying that I think this approach is just incredibly exciting, coming out of the fact that modified RNAs and lipid nanoparticles have been validated now in millions and millions of people in terms of safety. Now we can target these to specific cells in the body. And I think we'll be able to make CAR T cells targeting fibroblasts to treat a host of fibrotic diseases, perhaps to treat clones of B cells that might be responsible for autoimmune diseases, to treat senescent cells, to try to alleviate some of the disorders of aging where senescent cells build up. So we're eager to pursue this approach in the laboratory in a host of small and large animal models with an eye towards translating this to first-in-human clinical trials. And I'll stop there and thank my laboratory. That was the last picture before COVID of us getting together at a restaurant. In particular, I want to thank Haig Agajanian, who initiated this work in the lab, who's now moving over to Capstan Therapeutics to try to see the translation of this into human trials, and Joel Rurik, who's taken this work forward, especially the more recent mRNA lipid nanoparticle work that I showed you in the last few slides. Thank you very much for your attention, and I'm happy to take some questions.